Hello and welcome. Next to me I have the Tower of Dreams, created by Guozin Zhu in 1999. It represents a stairway to dreams, and behind it we have the office of the President of the National Shenggong University, President Jenny Su, located in the Yngping building. Now, my name is Torbjörn Nordling, and I'm an assistant professor in automatic control at the Department of Mechanical Engineering, and I'm the general chair of this workshop. And now it's my great honor to introduce our honorary chair, Chair Professor Wu, who will be giving his welcome address.
It is a pleasure to welcome all of you on behalf of the National Shenggong University in Taiwan. I'm proud that our team, the NCKU Parkinson's Disease Quantifiers, are one of the finalists in the OpenCV AI competition. They are also arranging this workshop. Now let us all learn computer vision and AI using the OAK-D camera together.
Hi and welcome. I am Esteban Roman, a PhD student in the Mechanical Engineering Department in the National Shenggong University of Taiwan. I am one of the speaker coordinators for this workshop. It is my honor to introduce both Evangelos-Marios Nikolados and Nikolas Adaloglou. Evangelos-Marios Nikolados is a molecular biologist interested in AI-enabled synthetic biology. He holds degrees from Harvard University, Imperial College London, and is currently a second-year PhD student at the University of Edinburgh. Nikolas is an electrical and biomedical engineer that is interested in the development of human-centered AI solutions. He is currently working as a deep learning PhD researcher applied to medical imaging. Together, they will present AlphaFold 2, the AI breakthrough in a bioinformatic perspective. Their tutorial will introduce some minimal biology background to understand AlphaFold 2 models. They will briefly introduce some key concepts regarding protein structure and function, such as protein structure level, protein domains, motifs, redisues, and turns.
Okay, so hi everyone. I'm Evangelos, one of today's two speakers for this presentation. So along with Nicholas, we're going to focus on AlphaFold 2 from bioinformatics perspective. And before beginning, we would both like to thank Torbjörn for the invitation and the opportunity to present here for you today. So this talk was inspired by a blog post Nicholas and I co-authored for AI Summer, and which has attracted more than 20,000 views in about 3 months. And of course, you can find it here in the link. So an overview for today's talk, I'm going to talk to you about in the first part, and I'm going to cover questions like what is a protein, what are the levels of protein structure, and then we're going to focus a bit on domains, motifs, and turns. I'm going to give you the intuition behind on why it's necessary to use multiple sequence alignment as an input for AlphaFold 2. And then I'm going to highlight the difference between protein structure and protein folding prediction. Nicholas, in the second part, is going to talk about the association of biology with machine learning model design. And he's going to cover topics like representing amino acid sequences as embeddings to feed as input in AlphaFold, self-attention and transformers, invariant point attention for AlphaFold's core self-attention module, and we're going to close with some remarks on whether the protein structure prediction is actually solved. So I'm going to start essentially our story with the central dogma of molecular biology, which describes the process by which instructions in DNA are converted into a functional product that is a protein. So as shown in the top image to the right, the first step is to produce a messenger RNA through the process of transcription. Now this RNA molecule carries all the information we need, which is then transferred to special cellular machines called ribosomes. Over there, the mRNA is translated into polypeptide chain, which is essentially a different name for a protein. Of course, the actual processes and even the flow of information in the central dogma are more complicated than this image. However, this is a good starting point to ask the question of what is a protein. So proteins are complex molecules that play many critical roles in an organism. They do most of the work in cells and are required for the structure, function, and regulation of different processes. So they're made of hundreds or thousands of smaller units that are called amino acids. Now, amino acids are essentially attached to one another to form a long chain or a protein. There are many known amino acids, but only 20 are encoded by the universal genetic code that you can see in the picture at the bottom down here. So genetic code essentially refers to all the valid combinations of nucleotide bases. Specifically, if we return to the top image on the RNA level, you're going to see that essentially our sequence is chunked into subsequences of length 3. Now these triplets are called codons, and essentially one triplet results in an amino acid. So after describing essentially this introduction into how proteins are made, we're going to pass into into the levels that make up essentially the structure of a protein. So to describe a protein structure, we use 4 distinct levels. Now, the primary structure or first level is simply the sequence of amino acids in a polypeptide chain. Now, as you can see at the top, we have a polypeptide chain. So here our chain is made of alanine, glycine, valine, threonine, and so on. The next level, the second level, is called secondary structure, and it is essentially 3D arrangements of regular repeated patterns of folding. Now the most well represented patterns here are alpha helixes and beta sheets, as again you can see in the figure to the top right. Our next level is the third one, and it's called the tertiary structure, and describes the overall 3D shape of a protein molecule. Another way of seeing this is that the third level describes the spatial relationship of the secondary structures to one another. Finally, we have the fourth level or quaternary structure that describes protein-protein interactions in closely packed arrangements. Now, what is important to remember here is that not all proteins have 4 levels of organization. Several proteins are functional and ready to go after completing their folding in the third level. However, others need essentially to assemble into larger complexes, and for that they need other proteins to come together. And these are the only ones that have essentially a 4th level. Continuing, we're going to focus a bit on different characteristics of a protein's structure, and we're going to talk about domains, motifs, and turns. Starting from domains, they are self-stabilizing parts of a protein chain. Now, what that essentially means is that a domain folds independently of the rest of the protein. That also means that if we were to cut it out from the protein, it wouldn't unfold and it would still remain functional. Now this is a very, very important property, because molecular evolution uses domains as building blocks, recombining them in different arrangements to create proteins with different functions. And this is also what we humans are doing with genetic engineering in many cases. So here at the top, I have used the AlphaFold database to get essentially the protein structure prediction for a transporter protein encoded by a human gene. And I have highlighted one of the two domains that make this protein. So it's essentially the one you can see in green. Now, if we are to zoom in a bit on a domain, another characteristic of structure is motifs. So motifs are 3D regions that are formed from connecting different secondary structure elements like alpha helixes and beta sheets. So to give you a better idea, I have rotated the protein on top and looked inside the domain to find one such motif, which is the one in the red circle. So here we have a motif that's called helix loop helix, and it is a very common motif occurring in different proteins. And that gives us also the chance to talk about very briefly about turns and loops. So turns and loops are also a type of secondary structure that are used to connect alpha helixes and beta strands. So as you can see, essentially in the part in the red circle, we have the helix, which essentially takes us away from the center of the protein, and then a loop allows us to change direction, allowing to fold back on the core protein structure. So having covered most of the structural biology that you would need to understand some of the terminology when it comes to structural biology and AlphaFold, we are going to continue with multiple sequence alignment, or MSA. Now this may seem daunting to many of you that are coming into biological problems without prior knowledge of biology, but MSA is essentially refers to a set of methods that have the goal of aligning 3 or more biological sequences of similar length. Now I'm saying biological sequences because we can use those techniques with either protein sequences, DNA, or RNA. So here we have an example at the top that essentially we are aligning, I believe, 8 different protein sequences. And if you start looking at the colors, you're going to see that in several columns we have conservation of a particular amino acid, or in other cases we have conservation of the property of the amino acids. Now one of the reasons we're using MSA comes from biological observation that people doing structural biology have uncovered by aligning millions of these proteins to each other, and it's a concept called coevolution. So to understand that, we are going to start from the bottom figure, specifically to the right, where we have this polypeptide chain, where each of the gray circles is essentially an amino acid. Now focusing on the 2 with the orange asterisks, the figure here essentially tells us that these 2 amino acids are into contact in the 3D essentially shape of the protein. What coevolution essentially tells us is that if for some reason we were to have a mutation on one of these essentially positions, then in order to preserve the structure of the overall molecule, a mutation on the neighboring position that is in contact with that with the first amino acid would have to be such that contact is again facilitated in the new amino acid in that position. So, and this is because mutations are occurring all the time, and especially if we think in the time scale that evolution works, so we're talking billions of years, then if every mutation was entirely random in these cases, we would end up with essentially losing function every time that a mutation occurred. So coevolution is essentially ensuring that even if a mutation occurs, then a mutation on the neighboring side is going to still facilitate contact of the two amino acids. So that essentially implies that structure in this case is more conserved than the primary structure of our protein. So, and remember, by primary structure, essentially we refer to the sequence of the amino acids. And this is why MSA is a very important input for AlphaFold. So by using essentially the different methods to do MSA, we can do this. We can look at this coevolution phenomenon over millions of different combinations. And finally, I'm going to close with a remark on the difference between folding and structure prediction. So many of you have probably read different articles talking about how DeepMind and AlphaFold solved the folding problem. So that is wrong. That is a very wrong statement, because AlphaFold is predicting the structure of a protein. So what the figure here is actually showing you is that if we start from the left, where we have the unfolded protein, in order for this polypeptide chain to assume its final 3D structure, it will have to go through different processes and stages that depend on energy barriers, conditions, and so on. So essentially, folding describes the process that will take us to the outcome, which is the 3D structure. And AlphaFold essentially is not predicting anything from what is happening in the middle, in the process, but predicts directly the 3D structure. And with that, I want to close my part and pass it on to Nikolas.
Hello. So after Evangelos presented some biology related stuff related to AlphaFold, I will talk about associations between biology and machine learning model design. So from the first overview diagram of AlphaFold, you can see that one can divide it in 3 parts. The first part has to do with the pre-processing stage and the search in databases for relative information. For example, to form the multiple sequence alignment, MSA in short, there's a lot of search in huge protein databases on the upper part, and as well as some other structural similar structures, proteins with similar structures that create some structural templates. So this process requires heavy domain knowledge, And from this stage, we have two output representations. In the upper part, it's multiple sequence alignment. It has a representation for itself. And in the second part, we have a pair representation, which uses both the multiple sequence alignment as well as the input sequence that we want to predict the 3D structure and the structural templates. The second stage refers to the transformer-based processing, which is the heavy processing. In this stage, the heavy protein representation refining is happening. And then we need the third stage, which does the actual prediction. This basically transforms the two representations that have been refined from the transformer architecture called EvoFormer, and it actually produces the actual output signal that we want, in this case the 3D structure of the protein. It's also worth noting that this step is here we have some recurrency, some feedback to the representation, which is usually 3 times that work best for the CASP competition. And this is a very key component for AlphaFold to work in the best setup. If you are familiar with recurrent neural networks, it's a kind of iterative process. And I also would like to say that these databases have a lot of bioinformatics information and a lot of domain knowledge that's associated with them. So let's start from the beginning. We have an input sequence, and we would like, in case of protein, residues or amino acids, and we would like to see how we can process it with a machine learning model and deep neural network that will be optimized with gradient descent. So the problem is that amino acids, similar to words, are discrete. We use symbols to show them, while in fact they are not discrete symbols. So to represent the input sequence, one has to project amino acids, all the discrete symbols, in a distributed continuous space of vectors that will be changed throughout training. So at the beginning of the training, these random vectors will not have any structure, but at the end of the training, some structure will be formed. For example, in text, as shown on the right, similar words will be grouped together. This is the idea of embedding, that it's not new. If you are familiar with natural language processing, it can happen in a word level, character level, amino acid level, or nucleotides for DNA sequences. After this step, we need to add some sort of order, because the transformer-based architecture have no notion of order, and that's why they are permutation invariant. So the idea is to add another vector that represents the position, which can be hard-coded like sinusoidal signs, or even another embedding. It's called, in this case, they're called positional embeddings. Then the heavy processing is happening in the EvoFormer transformers, and even though the AlphaFold architecture is highly specialized for 3D structure prediction of proteins, the core component is self-attention. Self-attention is a mechanism that can be regarded as a search in a database. For example, I've made an example with the video search in the database. First, we have the initial, the 3 key representations. The first one is the query. It's the user wants to search for something like a video. And then we have to use some distinct identifiers called keys that will do some kind of mapping between the query, what we want to look for, and the the keys in the database. So this will be translated in mathematics as a matrix multiplication, which is basically a similarity calculation. And it's where the highest part of the biggest computational issues arise with the self-attention mechanism. We will see then how AlphaFold use this idea to reduce the computational complexity in some parts of the pipeline. Then we know, after this multiplication similarity, we know where to look for. We have some kind of attention, we know where to look for, but we don't have the information that we actually want. So after you need to do another multiplication in mathematics, but in this example it's a mapping, which will translate the weights to the actual values that we want. And this gives us the relevant information. This is the principle of feature-based lookup in literature. So Selfintention is doing something very similar. In fact, here you can see the two multiplication. Q represent the queries, K represent the keys, and V the values. So we have two multiplications. The first one will be the similarity, and it will give us, after the softmax, the attention weights, where to look for on the database, while the multiplication with V will give us the actual information that we want. So AlphaFold, the AlphaFold model uses a special form of attention, of self-attention, which is called actual attention, in the processing of multiple sequence alignment, because as you have seen, multiple sequence alignment is kind of a 2D has a kind of 2D structure where rows represent similar proteins, while columns, in the column we have structure. We add all the similar proteins together. So to make self-attention more computationally effective, AlphaFold uses this actual attention mechanism, which reduces the complexity. How? First, we apply attention in a row-wise level between the rows, and this will give us relevant information for each sequence separately. And then in the second stage, we will apply attention only in the column-wise level. So this will combine the residue information from different sequences of the multiple sequence alignment. This idea was already familiar in computer visions from 2019 and 2020. Another intuition, not exactly from AlphaFold, but to associate machine learning, model design, and biology, is to use this idea from MSA Transformer paper, which proposes, again, since multiple sequence alignment have a big structure overlap, to share the information of the heads of the attention, the different representations of the attention, to share it between inside the self-attention module. So this also gains a lot of computational complexity. We save a lot of complexity. Then I will talk about invariance and equivariance. From those who are from a computer vision background, you probably know that CNNs are translation equivariant. Means that if you slide an image, an input image, the output features will be also slided. This is equivariance, while invariance is that independent of, similar to transformers, if you don't have any positional encoding, independent of the way you provide a sentence or an amino acid sequence, the output will be the same. So in a 3D protein structure prediction, we need some kind of invariance to rotations and translations that AlphaFold encapsulates in the core self-attention module. This is nothing more than a conventional attention, as you see in the red blocks. Here it's the keys with queries multiplication, which is the heavy lifting happens here, and the softmax operation. And after you see the another multiplication with the values. However, you see a lot of additional information to be added in different parts, either from the single representation of the protein of interest, the sequence of interest, or from the pair representation that usually fuses information of multiple sequence alignment. So with some very clever tricks, they managed to create some invariant mechanism to rotation and translation. So local updates that happening will not change the overall global 3D structure of the protein. Finally, I would like to say that AlphaFold uses Quaternions, which is an alternative representation instead of 3x3 rotation matrices as shown here on the left. So this representation of Quaternions creates a more smooth landscape for backpropagation to be optimized. It doesn't have edge cases, for example, with rotation of 0 degrees and 360 degrees to be the same. And it uses 4 numbers instead of 9 that a normal rotation matrix would have. And interestingly, they fix the first number to be 1 in the AlphaFold paper, which means that they want to predict small rotations. I find this representation, the usage of Quaternions, to be very important for the success of AlphaFold 2. And the final question is if 3D protein structure is solved, and as Dmitry Korkin said in his interview with Alex Friedman, where you can find in the link, AlphaFold 2 is amazing, but protein folding is not solved. And the reason he said this is that the competition that AlphaFold 2 has shown remarkable success is constrained to single or 2 domain protein domains, while the real-life proteins are multi-domain. And finally, there is no insight to the folding process. We only have a snapshot of the protein, of the final protein structure. You can reach us out on our emails, and we will be ready to answer your questions.
Hello, Mr. Adaloglou. Thank you very much for the great talk. I'm the host of today's Q&A session. We'll put the link of our Google Meet in the chat, so if any audience want to join us, please feel free to click it. At the same time, let me read the question in the chat for you. So there are some,
in the YouTube chat.
Okay. So there are some questions in both Twitch and YouTube chat. So let me read the first one into Twitch chat. Thank you for your great presentation. Why is the structure of protein so important? Could you give us some more examples?
Okay, I'm not the best person to answer this question because Evangelos has more knowledge in proteins. I'm just studying from a machine learning perspective. But the 3D structure is the most functional, the most important thing
for the function of the protein. You can see this pattern coming up again in human physiology, where the structure really determines the function. I don't have any specific examples because I'm not a biologist, but this concept, this pattern applies over and over again.
Thank you very much. The second question is, why recycling 3 times? Could it also be some automatic process?
Yes, so it's very strange to me also that this iterative process is necessary for it to work. If I would implement it myself in a lab and stuff, everybody tries end-to-end solutions. So I think it has to do with the transformer architecture that can refine the representation, even its own representation, with this feedback loop. After AlphaFold, there are many publications on transformers with feedback loops, but it's more for natural language processing.
Thank you very much. I see that we have some audience joining our Google Meet.
Okay.
So Sebastian, do you want to ask questions?
Yes, I have a question. I don't understand very well the final phrase of your presentation. Did you have said that this method only can catch only one snapshot of the folding process? Is that right?
Yes, exactly. The input is just a normal sequence of amino acids. There are 20 amino acids in humans, for example. And the output is just the coordinates, the distances in of the 3D shape of the protein. The distances, the relative distances. We have no information and no estimation about how this folding process. You can imagine, for example, in other problems of machine learning, like let's say image synthesis, where you try to generate an image, let's say from animals, but you have no idea of how this image is generated. It depends on the method that you will be using, but you can basically observe the output image, but you don't know exactly how this image is generated. AlphaFold is not interesting to predict the whole folding, because it's a much more complex thing to predict. They only try to predict in the first stage the stable structure, because there are a lot of jittering and folding, which is very difficult to predict.
So, do you have any idea how to advance or proceed in the direction to obtain the stable folding state? Maybe you can access to some kind of data set from the crystallization form of the protein. So maybe you can complete the different folding state with this information. It is possible? If it is that possible?
I'm not aware if there are databases with supervised data for the whole folding process, but maybe in the future they will try to see the. I think it's the most important step is to scale AlphaFold, not to make it as a model like bigger or anything, but to scale it to larger proteins. I think this is the primary goal. And yes, in the long term, maybe people would be interested to understand the folding process itself.
Okay. Thanks very much for your answers.
Thank you very much. You're welcome.
Thank you very much. We have also another audience join us. So, James, do you have any questions?
I think his microphone is off.
Yeah, it seems it's muted. Sebastian, do you have any more questions?
No, that's fine for me.
Okay. So while at the same time waiting for James, let me also read some question in the YouTube chat. So that question is, how does the model rank bioinformatics features that are more important than doing query search?
I'm not sure about the statement of ranking. In general, I would say that self-attention, in my experience, but also in a wider range of tasks in natural language processing and computer vision, self-attention is a very good mechanism to ignore information. So this kind of ranking that people asked, I think it comes from the realization that a lot of ignoring of the information, a lot of focus comes from the attention weights itself. This is the first part. But secondly, I would like to emphasize that AlphaFold uses different types from specialized self-attention. They didn't just apply the original mechanism. And does that answer the question? I'm not sure, but this is the only thing that I can say with some confidence.
Thank you very much. I think it's detailed explanation. So if audience want to give more information about that or want to ask more questions, please feel free to add more. The second question from the same person is how to know the exact position of the amino acid symbol to do the continuous vectors representations?
Okay, I think the question here has to do with the positional encodings of AlphaFold. From a first glance at the paper, we know that AlphaFold takes into account the relative distance between the amino acids in the sequence. And actually, they look at the 32 neighbors of the amino acid sequence. So this is how they encode the position information from the primary structure, from the first level of proteins. And they say some things that this can be very good to, since they are clipping the relative position encodings to just looking at the 32 neighbors. This is kind of an inductive bias that they don't want to emphasize in the distances and the position in the primary sequence level to let the model to enable the model to find the distances in the 3D structure, for example.
Hey, thank you very much. Next question is, what will be the next improvements of AlphaFold 2?
Yeah, so my answer is based on as I referenced in the end of the talk on Dmitry Korkin. Maybe I pronounced correct, maybe not. So the problem with AlphaFold and the CASP competition is that it solves quite well for small proteins, like 1 or 2 domain proteins. So the natural extension would be to scale it in terms of bigger proteins and see the challenges in more real-world proteins, let's say.
Thank you very much. The next question is so far the last one. It is, what is the major challenges of collaborating between biologists and engineers? What do you think is the biggest gap between them during the collaboration, and how to cover this gap from both sides?
Yeah, actually, I have a similar multidisciplinary issues because I'm usually working with experts from the medical field, and the pattern is more or less the same. To bridge the gap, usually the person who works in the machine learning world has to put some more effort to understand the terminology rather than expect the domain expert, whether it's a biologist, a clinician, or any type of domain knowledge person to put the effort to understand more stuff from your domain, because machine learning is a kind of umbrella term. It applies pretty much everywhere, and domain experts expect from you to bridge this gap, in my opinion. They will try to understand a bit, but it's usually the person who comes from the machine learning or deep learning domain that has to put the extra effort, in my experience.
Thank you very much. Sebastian and James, do you have some more questions?
Okay.
No, guys, I don't have any more question.
Okay.
Okay. Maybe there is another person in the chat who wants to ask something.
Yes, James, if you can hear us, and if you cannot use your mic, you can also type in the Google Meet. I'll try to speak them. While we are waiting, let me just mention that so tomorrow we'll have another four workshops. The first one will be at tomorrow 8 a.m. Taipei time, and we will later put our website link in our chat. So for more information, please check that.
Is there any other question for now?
It seems there's no question in the chat.
Okay, I think I didn't expect. I'm happy to see that people are interested, and it's nice that they asked so many questions. Maybe I couldn't answer them, all of them. I couldn't address them, all of them appropriately, but at least I tried to give some indications on where to look for.
Yes, thank you very much. I think all the answers are so valuable. I believe every audience learned a lot from you.
Okay, that's nice.
So I think this is, since there's no question pop out, I think this is the end of today's workshop. Thank you very much for your answer, and thank you everyone joining us. And look forward to seeing all of you in tomorrow's workshop. Thank you everyone. Thank you, Mr. Adaloglou.
Thank you very much. You're welcome.
Thank you. See you. Take care.
Enjoy.